Good afternoon, everyone. Once again, I want to welcome you to today's webinar. Before we get started, I have a few procedures that I want to go over. I would like everyone to please mute your line. I've taken care of that on the back end, but if you become unmuted at some point, please mute yourself. You can use the chat box to ask questions, and all questions will be answered at the end of the presentation. For any additional question not answered during the webinar, please email us at education@nola.gov, and there are no CEUs offered for this webinar today. Next, you'll hear Dr. Jennifer Bro introduce today's speaker.
All right, thank you. Can everyone hear me?
Yes, we can hear you.
Okay, great. It's a great pleasure today to introduce Dr. Stephen Juliano, my former PhD advisor and someone I've worked with for a very long time. Steve is a distinguished professor of ecology in the School of Biological Sciences at Illinois State University. Steve has a BA in biology from Kalamazoo College, a master's in entomology from Cornell, PhD in zoology from Pennsylvania State University, and he completed his postdoctoral research in ecology at the University of York in England. Steve's research interests include community, population, and behavioral ecology, applied statistics, and the application of mathematical tools to ecology. He uses container-dwelling mosquitoes as a model system, focusing on how processes in the aquatic environment influence community composition and adult mosquito traits. I'd like to say that his work on invasive species and container system ecology carries a lot of implications for those of us who work in vector control and IPM, and I think he's going to talk about some of that stuff today. I also want to mention that in 2019, Steve served as a panelist for the University of Illinois Critical Conversation on Genetically Modified Mosquitoes. And last year, he gave a webinar for the foundation of the NIH as part of the Gene Convene Global Collaborative, where he spoke about the ecological relationships of mosquito vectors and risk assessment for gene drive technology. So with that, we're really excited to have him here today. Steve, thank you for being here, and you can take over whenever you're ready.
Okay, thank you, Jennifer, and thank you to New Orleans Mosquito Control for the invitation. Let me share my screen and make sure you're seeing everything. And are you seeing my opening slide right now?
Yes, we are.
Okay, and you hear me fine?
Yes, you're
Hear you good, Steve.
Okay, so thanks to everyone for coming. And as Jen said, my work for all of my recent career has been on ecology of container mosquitoes, particularly Aedes, and a lot of that has involved competition among Aedes, central to what I'm going to talk about today. Those of you who know my work also know I'm not someone who works in mosquito control, but I do think that the ecological processes that I've studied, particularly competition among Aedes larvae, have some real important implications for the application of mosquito control. And that's what I'll start to tell you about right now. I don't have to remind this audience about the importance of container-dwelling Aedes species as vectors of viruses that affect humans. This is a short list of some of the major viruses that are transmitted by container-dwelling Aedes. And they're all of them health problems of greater or lesser extent. Yellow fever is highlighted for one reason. It's evident in this picture down here, which is from a couple years ago when there was a yellow fever outbreak in Central Africa. And it shows the response to that outbreak, which was a massive vaccination campaign locally. 30 million people vaccinated, like this brave little girl down here. By the way, are you seeing the pointer?
Yes, we can see it.
And yellow fever is, of course, the only one of these vector-borne viruses for which we have an effective, widely available vaccine. For all of the other viruses transmitted by mosquitoes, it is mosquito control that is the principal tool for public health fighting off these illnesses. And so the success of mosquito control becomes very important from a public health perspective for all of these cases. So mosquito control, simple in principle. You impose a source of extrinsic mortality on the mosquito population. I tend to think of this mostly as imposing that on larvae, the aquatic stages of mosquito life cycle. And in doing so, what you hope to do is reduce the density of the mosquito population. And the goal of that in a health context is to produce what might be characterized as density mediated effects on disease transmission. That is, by reducing the density of the vector, you would try to reduce the transmission of the disease. A side effect of that reduction in density, though, is that you will reduce intraspecific competition among the mosquitoes that you are targeting. And that has other consequences. Those consequences could be grouped into two categories. One, trait-mediated effects. Effects on the population of individuals that is the target and their ability to transmit disease. If you want to think through a clear example of a trait-mediated effect that might arise from mosquito control, consider the size of the adults. You can measure the size by wing length, and what you see in this graph is just an example of how, as you raise the density, you decrease the size of the resulting adults. And if you imagine reducing the density, you see the opposite would happen. If you go from a high density situation to a low density situation, you get bigger adults. The other thing, and I'll talk some about the possible consequences of trait-mediated effects of mosquito control, but density-mediated effects are the goal of mosquito control, and they're also more complicated than we tend to think. And I'll talk more about that as this talk goes on as well. Oh, there it goes. Okay. So envision population of container-dwelling mosquitoes like Aedes albopictus, where they're crowded to some degree. The larvae are forming a dense population in the aquatic environment. That crowding will, like I noted earlier, produce smaller adults, but there'll be other things that change too. The adults from these conditions will usually be delayed in their emergence to the adult stage. If you intervene by doing something like, let's say, biological control by adding a predator, and that predator reduces the density, the survivors, if there are any, will benefit from that reduction. They will develop faster, reach the adult stage sooner, and be usually considerably larger. Beyond those really simple life history effects, though, there are some potentially trait-mediated effects of density changes produced by mosquito control that might affect disease transmission in a very direct way. Vector competence is a measure of the probability that a female mosquito becomes able to transmit a pathogen after taking an infectious blood meal. And it is a product of the mosquito's physiological state. And mosquitoes erect both physical and immunological barriers to the pathogens that they might ingest during a blood meal. So in this diagram, you see a cross-section of a mosquito. The female takes blood into her gut. If that blood contains a virus, to make the mosquito infectious, those viruses have to infect the gut and propagate. They have to break out of the gut, escape the mid-gut escape barrier, and infect the rest of the body, propagate further and invade the salivary glands, infect those tissues, and escape to the lumen, where they can finally be transmitted by a subsequent bite. The mosquito females invest in defenses at all these stages, physical and immunological barriers to inhibit the virus, simply because the virus is by no means good for the mosquito. It is exploiting that mosquito. Now, vector competence, that probability of an individual becoming infectious, contributes to another concept, vectorial capacity, which is a population characteristic for the vector population that quantifies the number of infective bites per person per day the population of mosquitoes can produce. And it has multiple components, some of which are directly related to the outcomes of mosquito control. I already mentioned vector competence, that's here as V, but M is simply the number of biting females, and that is of course directly related to how many adults are produced from the population of mosquitoes that are the target of mosquito control. There's the proportion that bite, and also another important characteristic of this vectorial capacity equation is the daily probability of female survival. That is simply because all of the processes of escaping the barriers erected by the mosquito to the virus and the virus getting to the salivary gland takes a certain amount of time, the extrinsic incubation period, symbolized by N. And if the mosquito doesn't live long enough for the virus to reach the salivary glands, it will not be able to transmit. My colleague Barry Alto did a really fascinating experiment with Aedes aegypti and Aedes albopictus, investigating how competition among larvae affects vector competence as a component of that vectorial capacity. And so what he did was set up three treatments. One was each of the two species alone at low density, shown in the open symbols for albopictus and aegypti, and then alone at high density, shown in the black for the two species, where they will be subject to intraspecific competition for food. And then the two species together at the same high density, where they will have both inter and intra-specific competition for food. And then Barry raised the adults under those conditions, got the adult females, offered them a blood meal infected with dengue, and then determined after an appropriate incubation period what fraction of those individuals produced an infection and what fraction of those individuals produced a disseminated infection, which is simply a measure of whether that dengue virus had escaped the gut of the mosquito. And what you see is for the two species, there was a clear effect of competition on vector competence by either of these measures. Particularly for albopictus, there was a really strong, highly significant effect where when reared at low density, and lacking much in the way of competitive interactions, the proportion infected was significantly lower, and the proportion with disseminated infection was significantly lower than it was for those that were subject to inter or intraspecific competition. That same trend was present in Aedes aegypti, although it was not as strong and not, strictly speaking, significant. And I'll point out this little insert down here that shows the other thing that I noted in the previous slides, that this set of treatments also affected the size of the adults that were produced. At low density, Barry was getting large adults that developed quickly. And at the high competitive densities, the opposite, small adults that developed slowly. So this says that competition in the larval stage can have a prominent effect on vector competence in the adult stage, and it would appear that it enhances the likelihood that the mosquito becomes infectious. That's the effect of competition. That might be due to defending themselves against the virus being costly. Well, this is a study that was done by Jen when she worked in my lab, also with Aedes aegypti, on a different aspect of how intraspecific competition among larvae might affect a component of vectorial capacity. She raised Egypti larvae at either high density or low density in standard conditions, and then got the adults and exposed them to microfilaria of a Brugia nematode. And I'm not going to really talk about the Brugia in this case. What I do want you to notice is these survival curves. These are the proportion of the initial number of adults that were alive a given number of hours after they closed. And what you see is a strong and significant effect of the density at which they were reared on the longevity of the mosquitoes. Those reared at high density live considerably less time than those at low density. This might also affect vectorial capacity. And if you go back and think about that expression, this would have the opposite effect on vectorial capacity. Competition would lessen the vectorial capacity. So I've just talked about two trait-mediated effects of competition among larvae on the components of vectorial capacity that go in opposite directions. Those could be characterized as leading to two hypotheses. On the one hand, the competition longevity hypothesis would suggest that competition among larvae results in small female mosquitoes that, based on longevity, should produce a lesser frequency of dengue infection in a population of adults in nature. So you'd expect that as female adult size increases in a natural population, that the probability of seeing infection among the females would be increased as size increases if this competition longevity hypothesis is right. If you think, though, about the competition susceptibility hypothesis that arises from Barry Alto's work, you might expect the opposite pattern. Competition should produce small adult females. Those are less able to defend themselves against infection. And you might expect, if this hypothesis is prominent in real life, you would get a declining relationship of frequency of dengue infection to female size. So which of these really takes place in nature, if any? We were interested in this, and we being me and colleagues at Osvaldo Cruz in Rio de Janeiro, Brazil, investigated the relationship between frequency of dengue infection and adult size for field-collected Aedes aegypti. This was during a major outbreak of dengue in Rio de Janeiro in 2008. And you see the data on the number of human cases of dengue in Rio. It was a massive outbreak. We sampled adult Aedes aegypti all over the metropolitan area of Rio de Janeiro at all these pinpoints, got the individual females that were collected, brought them back to the lab, measured the wings of the females to get a measure of size, and then screened every individual for dengue infection. And then asked the question, in this population, is the frequency of dengue among the females increasing with size or decreasing with size or neither? And the answer is here. This is the fitted regression function to these data. There were 545 females collected. You can see that the vertical axis is the regression prediction of the frequency of dengue among those females. The wing length is on this axis, and the day of collection across the months of dengue outbreak is across this axis. And the result is very clear. There is a monotonic, significant increase in the frequency of dengue infection as the size of the female mosquitoes go up. This is clear evidence that that competition longevity hypothesis may well be the dominant feature in the field. This doesn't necessarily mean that the vector competence hypothesis is wrong. It just may mean that the effect of longevity on likelihood of becoming infectious is the much stronger component. So trait-mediated effects seem to be important in the field for these mosquitoes during a real-life dengue outbreak. Okay, I want to shift from talking about the trait-mediated effects that might arise due to larval competition and density, and talk instead about the effects of intraspecific competition and broadly density-mediated effects, and some of the paradoxical effects that may result from the interaction of larval density and processes like mosquito control that reduce the larval density. And this brings me to a description of the Hydra effect. And those of you who remember your Greek mythology will recognize this vase as showing the mythological Hydra and Hercules and his battle with it. And if you remember the story, the Hydra had a characteristic that if you cut off one of the heads of this monster, two heads grew back in its place. And the Hydra effect specifically is defined as a case where the equilibrium density of a population increases with the imposition of mortality on that population. Now that's in many ways counterintuitive, but what we think is going on to produce this kind of effect is that the mortality reduces the density and reduces the detrimental effects of intraspecific competition on survival of individuals. So any survivors that are not subject to mortality benefit so greatly from the density reduction that you actually get more individuals out. The theoreticians, and I'll recommend this paper by Peter Abrams, that have studied this have predicted several things. One is that for this Hydra effect to occur, density dependence has to yield overcompensation in response to mortality. That means that in a single generation, the number of adults that are produced from a larval cohort must at some point increase as the proportion killed increases. And you see that in this hump-shaped part of the curve. We expect that it wouldn't be uniform, that at some point you reach a high enough rate of killing that you would depress the number of adults produced. But this overcompensatory response in the short term has to occur for the Hydra effect to occur. The second thing that has to happen for both overcompensation and the Hydra effect is that the extrinsic mortality is predicted to have to act before the action of density dependence and intraspecific competition. So what you would get from this, particularly this last item, is that the earlier in the larval life mortality acts, the more likely you are to get overcompensation, and the more likely you are, therefore, to have the Hydra effect, that increase in equilibrium density, occur. So this is a general prediction about populations. It's not specific to mosquitoes by any means, but we were interested in testing this timing hypothesis. That is, the hypothesis is that when mortality acts before intraspecific competition, overcompensation and the Hydra effect are more likely. The prediction is that if you vary the timing of mortality, the earlier you cause it, the more likely you are to get overcompensation, all other things being equal. And this is work by one of my PhD students, Chris McIntyre, who put this to the test in a lab experiment with Aedes albopictus. And this is how the experiment was designed. We had a standard cohort of 250 newly hatched Aedes albopictus larvae with standard rearing conditions in a standard sized container with a standard amount of food. In the controls, we did nothing. Over time, some of those mosquitoes would have died due to crowding and intraspecific competition, and some of them would have made it to adulthood. And that's the question marks there just show we didn't do anything to control the densities after that initial condition. In early mortality treatment, on day 2, we imposed 48% mortality on the population of larvae by randomly removing 48% of the individuals. And then we left them go, however they continue to survive, grow, and develop. In late mortality, we imposed the same 48% mortality, but only on day 8. And you see that we held the density constant by replacing individuals that died any time before we imposed our mortality. So we really tightly controlled the density all along, only imposing mortality on day 8. And then the fourth treatment was what we called repeated mortality, where on day 2, 4, and 6, we randomly removed 20% of the existing population each time, which resulted by day 6 in the same cumulative removal of 48% of the population. And our question then is, which of these is most likely to lead to an overcompensatory response, that is more surviving adults than the control? And ultimately, which of these is more likely to lead to the Hydra effect? And these are the results that Chris got. You can see the statistics down at the bottom for adults combined, for male mosquitoes, and for female mosquitoes. And what you see is they're all statistically significant. And particularly for females and for all adults combined, the main result is that, exactly as predicted, early mortality led to a significant increase in the number of adults produced. So you can, in this case, kill off almost half the population and actually get more surviving adults than if you had done nothing. You'll also notice that for the other timings of mortality, repeated or late, we didn't really produce a significant reduction in the number of adults produced. There was about the same number in all cases, statistically anyway, indistinguishable from control, so that any source of mortality might not produce overcompensation, but what would be characterized as compensation, where you impose mortality and the end result is no different than if you had done nothing. So that's about the overcompensatory effect, the production of adults. That's a bit different from the Hydra effect, which is all about the equilibrium population that would result from that. And that goes to the population's rate of growth. And so from this experiment, we wanted to get an estimate of the rate of increase for the different cohorts of mosquitoes we were raising. And there's a published demographic tool called the Index of Performance. This is from work by Todd Livdahl and George Sugiura. We have used this a lot to estimate cohort rates of increase for experimental mosquito populations. I'm not going to go into the detail, but suffice to say, it's derived from life table statistics, and it makes use of things like the initial number of females, the number of surviving females, the time it took them to reach adulthood, and the sizes of the females, which is used as a predictor of eggs laid by females. And we use that to get an estimate of the rate of increase for the cohorts of mosquitoes in that experiment. And the results are shown here, and they show, not surprisingly, that again, the main effect of mortality was to produce a significant increase in the population's growth rate, estimated growth rate, with early mortality compared to control, or indeed compared to repeated or late mortality. Now, how does that relate to the Hydra effect, that equilibrium population? This is a theoretical plot of per capita growth rate on the vertical axis with zero growth over here, and initial density over here on the horizontal axis. And what's plotted is the expected relationship between those two things if a population followed logistic population growth, the standard ecological model with a carrying capacity. And the carrying capacity is simply where that line of growth rate crosses zero. This is the density at which the population has zero growth. That is the equilibrium density. So in an experiment, if you have a certain initial density and you estimate R prime, this index of performance, and then you have another group under the same conditions that has been subjected to some amount of mortality, and you see that the estimated growth rate is greater with mortality, that's including the mortality, then what you would guess is that, yes, the carrying capacity, K, has gone up. It's further out on this axis. That says that this increase in estimated rate of increase suggests the Hydra effect would be occurring in these experimental cohorts. And that's all the product of the interaction between the density and the density reduction and the potential benefits to the survivors that the reduction produced. Well, what I've told you is that it seems like when you set up a controlled experiment in the lab, you can produce conditions that make it seem like mosquitoes would show overcompensation in response to mortality and this Hydra effect. Well, that's interesting, but what's maybe more interesting is how likely is that to happen in the field, right? It's one thing to show it's possible. It's another thing to show that it might occur in nature. This is work by my current PhD student, Kate Evans. And she did a set of field experiments that addressed this likelihood of overcompensation in the field. What she did was put some containers out into a natural environment, fill them with water, and allow them to collect leaf litter and dead insects and other detritus that fuels bacterial growth, and to be colonized by whatever populations of mosquitoes, particularly Aedes, were present. And then after 6 weeks, we came back and removed and counted all the colonists so that we had an idea of the typical density of mosquitoes at the site. And then we stocked those containers with their resources with standard densities of Aedes spanning the observed range of densities we saw in the field, going from low to high. And then we sealed the containers and returned them to the field and came back after 14 days and counted the survivors. And what we're looking for here is how does survival of individuals in these containers relate to the initial density? So there's no mortality being imposed, but what we are looking for is the relationship between survival and density. Here's a set of relationships that are described by this expression, where S is the number of surviving individuals and H is the initial density of hatchling mosquitoes. And I'm not going to go into the meaning of all these parameters in the equations, except this one, D. D is the one that really controls the shape of this relationship. If D equals 0, you would expect a straight line relationship. As density goes up, survivors goes up. If D is between 0 and 1, you expect this kind of decelerating relationship where survival is going up, but at a slower and slower rate. If D is equal to 1, you expect this relationship, where as initial density goes up, the number of survivors approaches an asymptote, a maximum. And if D is greater than 1, you expect this shape, where the initial density, as it increases, produces more survivors, but eventually the reverse happens. At higher and higher densities, fewer and fewer survivors are produced. So that's what D controls in this expression. Why is that important? Well, here's that kind of hump-shaped curve that I was just showing you for number of survivors versus initial density. If you envision starting at some point like this one at a high density and imposing some mortality on the population at that density, you reduce it down to this density, you might expect to get more survivors if you're on this right-hand side of the curve. That is why we say that this hump-shaped curve is the signature for overcompensation in these field populations. And that's what we're looking for in the analysis of those data. Well, here is a set of results for that experiment done 2 times in 2 different sites, one in Missouri and one in Illinois, and plotting again the cumulative survivors at the end versus the initial larval density. Now, the little bar graphs at the bottom show you the distribution of actual number of colonists in each of these containers, just to show how these densities we used kind of span the range of what we saw in the field. In Missouri, when we did this, we got a value of D that was giant, 5.6, clearly significantly greater than 1, and indicative of this being a situation where we would expect overcompensation in response to mortality at these high densities. When we repeated this in a little bit different environment with the same species of mosquito in a cemetery in Illinois, we got a little bit different result. We got a D value indistinguishable statistically from 1.0. It's actually a little bit less than 1, but it's not significantly different from 1, although it is significantly greater than 0. This would predict compensation. That is, at these high densities, you might see a reduction in the population due to mortality, yielding very little change in the number of adults produced. So variable outcomes for the same species in two different contexts. When the experiment was done in a different location, southern Florida, with a different species, Aedes aegypti, we got, again, quite variable results. This was done in a suburban environment, basically in people's backyards. And in the early part of the season, the results are shown on the left. In the later part of the summer, the results are shown on the right. And what you see here is in the early season, we got, again, a case where the relationship of survival to density would predict compensation in response to mortality. In the later part of the season, when the densities of the mosquitoes were actually lower, which is a common thing for Aedes aegypti in Florida, you would get what you might characterize as a sub-additive effect. This was a very poorly fit regression, not significantly different from 0 for a slope. So additive, maybe sub-additive mortality. But again, kind of variable results depending on what circumstance we used, but at least one case where you might expect imposing mortality to maybe not yield the benefit that you might have thought. Finally, we did this with Aedes triceriatus 2 times in the same location in 2 different years. One time we got a clear signature for overcompensation with a very large D, significantly greater than 1. The second year we did it, we got another case of apparently additive mortality where D was not significantly different from 0, and you literally expect just a linear increase in adult production. So what do we take from all of this? One of the keys to this is that in these field sites, we get all kinds of variability in what you expect, but you get some circumstances where you would clearly expect overcompensation in response to mortality imposed on the mosquito population. Some cases where you might expect compensation, you might kill a substantial fraction of the mosquito larvae and get no change in the adult production. And you might get some cases of additive mortality. What this shows then is the effects of intraspecific competition globally, thinking about both the lab experiments and what we saw in the field, and the combination of competition and density reductions might affect multiple ones of these components of vectorial capacity. It might reduce biting number of females, but it might also increase them if overcompensation occurs. It might affect vector competence. The data from Rio de Janeiro suggest that reducing the density might actually make mosquitoes less able to become infected. That's that increase in dengue infection with size, which is probably mostly due to this probability of daily survival. Longer lived adults, larger adults, being more likely to become infected. A main takeaway from this is if you're interested in suppressing disease, there's a lot that goes into this expression that describes vectorial capacity that is going to be altered by interventions that reduce mosquito density. And it means that to do effective mosquito control, you have to take into account, I think, the impact on vectorial capacity, which is a product of both trait and density mediated effects that are produced by an intervention. And seems that it is both context, that is initial density dependent, and species dependent. I didn't really talk about that much, but we have other data that suggests that different species respond very differently to densities. What it means is that successful mosquito control is likely to require both species and context-specific understanding of the population you're targeting. Ideally, it seems like you would like to impose mortality that acts after the action of density dependence, late in larval development. And the lab experiments seem to suggest that killing something more than 50% of the population of mosquitoes in a larval aquatic environment is going to be necessary to really inhibit the population of mosquitoes. Now, in thinking about the practical implications of mosquito control, one thing this means is that if a particular intervention has a characteristic timing of mortality, it might affect how successful it's likely to be. And the two that I have highlighted here are interesting in this regard. Sterile male technique is a way that people are contemplating controlling mosquitoes and attempting to control mosquitoes. By its definition, sterile male technique, where you release males that have been sterilized in some way, they mate with females, and those females produce inviable eggs. This imposes early reductions in larval density before even the first instar arises. That early mortality seems likely to produce overcompensation. In contrast, some transgenic approaches, for instance, RIDDLE, which stands for release of insects with dominant lethal alleles, produces mortality typically in the fourth instar or pupa. And that may have a very different result because this is mortality that's more likely to be imposed after the action of density dependence. And each of these interventions may also affect disease transmission in unexpected ways because of those complicated effects of the different components of vectorial capacity. Well, I've talked a lot about intraspecific competition. I want to say a bit about interspecific competition because that's important among container Aedes as well. This has been exceptionally well studied for Aedes aegypti and Aedes albopictus, which are global invasive species, global vectors of major importance, and co-occur in many instances and compete intensely in some circumstances. So let's consider using a species-specific form of mosquito control, like sterile insect technique, or Wolbachia, another newish way of approaching mosquito control, where Wolbachia, the intracellular prokaryote that manipulates the insect host, can induce sterility, can induce shortened adult life, can do a variety of things to the mosquito. I mentioned RIDDLE earlier, and gene drive mechanisms are another way of trying to suppress mosquito populations in a species-specific way. All of these involve releasing, usually males of one species, to produce a reduction in the adult population of the target species in the wild. So let's think about these species-specific forms of control and what they might do to mosquitoes that are engaged in interspecific competition. Now, I will say right off the bat, there's a reason species-specific control of mosquitoes might be a great thing. Many of the detrimental effects of chemical pesticides arise from their non-target effects. You spray for mosquitoes or anything else for that matter, and those insecticides that are used don't just affect the target organism. They may affect many other insects and indeed many other organisms. And some of the early writing about the role of insecticides in particular was about the non-target effects of pesticides on things like caterpillars that are the food of birds and the carry-on effects that occurred. So if we're going to control mosquitoes, species specificity seems like a good idea because it allows you to minimize those non-target effects. But what are the consequences of suppressing one species if it's competing with another also important vector? So suppress one of these two vectors, what happens to the other one? Will species-specific control by population suppression result in competitive release of the non-target organism, enhancing the abundance of the non-target competitor if they co-occur somewhere, or contributing to invasion of new areas by a non-target competitor if you remove a species that's already present? A bunch of questions arise from this, particularly in the context of Albopictus versus Aegypti. What's the mechanism or mechanisms of competition? Is competition important in the field? Do these two mosquitoes coexist, or do we see competitive exclusion? And probably the most fundamental one of these is, would one species occupy the other species' niche if it wasn't there? And I'll define two things for you. The fundamental niche of a species describes the set of conditions in which a population persists indefinitely in the absence of any biotic interactions, competition or predation. That means that those environmental conditions in that fundamental niche are suitable for that organism when it's alone. The realized niche is the set of conditions in which a population persists indefinitely with biotic interactions. These you would think of are the set of conditions where that species is the best competitor, let's say, for the environmental circumstance in which you found it. When we think of the niches of these two mosquitoes, kind of the quick version would go like this. Aedes albopictus tends to be maybe rural to suburban in its distribution, tends to be a generalist feeder, biting a wide array of hosts, tends to be exophilic, mostly seeking blood meals outdoors. In contrast, Aedes aegypti tends to be thought of as an urban mosquito, often found in cities. Tends to be anthropophilic. There's some really good data showing it has a definite taste for humans as a blood source. And in many parts of the world, it is endophilic. That is, it will bite and indeed mate and oviposit indoors. So sounds like two different niches. To what extent might they move into the other's niche when given the opportunity? About the mechanisms and importance of competition in the field, there have been a number of studies, field experiments that have investigated this. I'll just mention one right here. This is an experiment from Rio de Janeiro done by Daniel Camara, where he manipulated the density of Aedes albopictus across a range and looked at its impact on Aedes aegypti. So put out a number of containers in the field, let them be colonized, and then removed the colonists and stocked them, those containers, with either a low density of Aedes aegypti alone, no competition, what he called a field density, combined Aedes aegypti and Aedes albopictus at twice the density here, or a high density, which is Aedes aegypti plus Aedes albopictus with albopictus at 2 times the density of the aegypti. So essentially 3 times the density as that you see here. And then looked at the production of survivors in the course of this experiment. And the result was a significant impact of competition across this range of typical densities that you might see in the field. And the same was true in 3 different seasons in Rio de Janeiro, autumn, spring, and summer. So it looks like competition among the larvae is pretty important, and Albopictus definitely has a detrimental impact on Aedes aegypti in this circumstance. And there have been other experiments that have shown this as well in other locations. There's another mechanism of competition that occurs between Albopictus and aegypti. This has been work largely done by my colleague Phil Lunabus. Saterization is reproductive interference where males of one species will inseminate heterospecific females. And because female mosquitoes tend to be resistant to mating after they have been inseminated, these females that have been mated by the wrong male will produce inviable eggs. And Phil showed that many Albopictus populations effectively saterize Aedes aegypti populations. And this too is a mechanism by which one of these species can inhibit the other. That acts in the adult stage. What about this coexistence question? What do we see when we look at the distribution of these species in nature? Well, first, you often see them coexisting. These are data from both Brazil and South Florida from a paper by Marita Brox. And what you see is along a gradient from urban through suburban to rural areas, aegypti dominates in the urban environments. That's this horizontal axis. The two are kind of co-equally abundant in suburban areas, and Aedes albopictus dominates in rural areas. So they frequently coexist. And even though you might find areas dominated by one or the other, those areas are often quite close to one another, so that emptying out one of these areas of the dominant species might lead to an influx of the non-target organism into that area if they find it suitable. There are other things that can contribute to coexistence. We found repeatedly in a variety of locations that we tend to find Aedes aegypti dominant at times and places that are drier, and Aedes albopictus tending to dominate at times and places that are wetter. We've seen this in Florida. Others have shown it in other parts of the world, including Africa. Some other more esoteric things, the nutrient content of detritus seems to influence coexistence, where you tend to get more coexistence when you have a high ratio of nitrogen to carbon, and more albopictus when you have a high ratio of carbon to nitrogen. Phil and his colleagues have found variation in saterization, where some populations of aegypti are resistant to being saterized, and some populations of albopictus are ineffective at saterization. And another one that's been studied a bit is aggregation of larvae. If larvae of the two species are aggregated in containers, they tend to encounter conspecifics mostly, and that reduces the impact of interspecific competition. So that can produce or contribute to coexistence. This may be the most important thing I want you to take from this part about interspecific competition, is this realized niche expansion. These are data from this paper on Aedes albopictus in Rome, where it is the only container-dwelling Aedes. There is no Aedes aegypti at the time this study was done. And what you see here is collections from urban and rural areas in the vicinity of Rome, and looking at the frequency of human versus non-human blood meals. And for albopictus, what you see in urban areas is they clearly will feed very extensively on human blood. Although they don't do that in rural areas to the same extent, they are opportunists, and they are quite happy to take human blood meals. Even more interesting is this. Aedes albopictus blood meals in Rome for individuals collected outdoors in blue and indoors in orange. And what you see is that in urban environments, Aedes albopictus seem to be quite happy to blood feed indoors in that urban setting. Even in some suburban areas or rural areas, they take a substantial number of blood meals apparently indoors. What this suggests to me is that albopictus anyway, given the opportunity of an empty niche, let's say a niche that was emptied out by effective control of Aedes aegypti, will very happily be an urban mosquito acting like Aedes aegypti. So they can, it appears, occupy each other's niche. What might happen with imposing species-specific mortality on competing populations of these two species? This is work done with individual-based modeling by my student, Kate Evans, where she built an agent-based model that had larvae competing for food in the aquatic environment, growing, developing, and maturing, and then coming out as adults and laying eggs. And then in the model, mortality early in the larval life, as might be imposed by sterile insect technique, was implemented at varying levels, ranging from 0 on up to 90% of the eggs failing to hatch. And what the models showed is quite interesting. If the mortality is specifically targeted at the superior competitor, which might be Aedes albopictus, that early acting mortality at low levels will lead to a slight increase in the abundance of adults of the target species here. That's the overcompensatory effect in this dark symbol. But more interestingly, as you ramp up that mortality to 25, 50, 75, and 90%, what you see is the non-target species, the poor competitor, increases greatly, even as the target is suppressed. It's a basic competition experiment, removing the better competitor. Well, what if you target the inferior competitor? Interestingly enough, it produces that similar effect. You reduce, as the mortality rate increases, the abundance of adults of the inferior competitor, but you increase the abundance of the superior competitor, which is pointing out the truism that competition, even with an inferior competitor, is not good for the good competitor. It's reducing its population. And if you remove a competitor, you are likely to benefit a non-target species that it competes with. So back to the questions that I posed. Mechanisms of competition, there are at least two, some among larvae, reproductive interference acting among adults. These species negatively affect one another. Is competition important in the field? Multiple experiments show that larval competition is important. I didn't have time to talk about it, but field data from Phil's lab suggest that saterization is important in some field sites too. We can see cases of coexistence, and we can see apparent cases of competitive exclusion for these two. But even when there is competitive exclusion, it's often true that the two species are near neighbors. They may be spatially segregated, but they can certainly be close enough to invade each other's habitat if that habitat is cleared out. And can they occupy each other's niche? There are numerous examples, and I gave you one, where we find that they do, where they can move into the environment that would be thought of as characteristic of the other species. What does this mean for species-specific mosquito control? This, like I said, there are obvious advantages to it, but we have this problem for these two species where you might inadvertently benefit the non-target, which would most likely be Aedes albopictus. Well, one solution might be, instead of thinking of population suppression, you might think about what's referred to as population replacement, where genetic modified mosquitoes have traits that induce resistance to the pathogens that they transmit. Some Wolbachia do this. Some investigators are working with genetic modifications that would produce refractory mosquitoes that would be unable to transmit viruses. This would have the advantage of reducing disease transmission, but leaving the population of mosquitoes in place to inhibit other species from coming in and taking over. You might think about a paired strategy where you might pair species-specific control with auto-dissemination, a really nifty new approach where mosquitoes that are released might carry with them very small amounts of insect growth regulators like pyriproxyfen that can be transferred to the aquatic environment. And there are some data that suggest pretty strongly that the amount of these growth regulators that can be transferred from an individual to the aquatic environment can be enough to inhibit the development of larvae. And so you'd have maybe sterile insect technique that controls a target species and auto-dissemination putting a little bit of a damper on non-target organisms that occupy the same habitat. Another approach might be to target for, let's say, SIT, both species, Albopictus and Aegypti. This is very sensible, but this, of course, would double the cost of doing the rearing operation to implement SIT. I have a lot of people to thank. Most notably, my students at Illinois State University, graduate and undergraduate, and a couple of postdocs as well, without whom I couldn't have done any of the work that I've done over the years. And I have been really fortunate to have a great batch of students that worked with me. I've had a longstanding collaboration with people at Florida Medical Entomology Laboratory, particularly Phil Lunibus, but others as well. The Brazil work was a product of collaborating with numerous researchers at Fiocruz. Some of our work was done at Tyson Research Center, some of it at Graceland Cemetery in Decatur, and with the cooperation of Macon Mosquito Control in Decatur, also at Parkland's Foundation in Illinois. And besides Illinois State University's research funding, I've been fortunate enough to be funded fairly frequently from National Institutes of Health for work on insect vectors of disease. And with that, I will stop and be glad to try and answer whatever questions you may have.
Thank you for that talk. The first question in the chat is, so in regards to overcompensation, do we know how field populations of generalist predators like copepods, tocs actually affect prey species? In my anecdotal experience, I will frequently come across containers with tocs and a decent amount of prey larvae despite predator presence.
Yeah. That's a really good question. And I'm very, very glad you asked that question, because I just happen to have one other slide that I wasn't going to talk about, but I will now, that answers that question, at least in part. It's very clear that most biological control agents, like predators, are maybe not terribly effective at eliminating their prey. They might be effective at reducing their prey. And I've had the same experience that you have had. You can often find a decent number of mosquito larvae, particularly Aedes larvae, in containers with Toxorhynchites. That's particularly true for Aedes triceriatus, which seems to do okay with Toxorhynchites compared to some of these invasive mosquitoes. But this is a slide that is more directly related to your question. Let me change something here. Bring this to full screen. This is an experiment done by another student in my lab, Zoe Neal, comparing the effect of the kind of controlled mortality we had been doing with a real live predator, namely Mesocyclops, cyclopoid copepod, that you see down in the lower left. And she did this with Aedes albopictus, Aedes aegypti, and Aedes triceriatus as the targets. And what we were interested in was two things. One was how the percent kill that we imposed on day 2 affected the production of adults. Because you remember the curve I showed you was that as the percent kill goes up, you expect eventually the number of adults produced to go down. But then we were also interested in, if you had a predator that was killing the same fraction, would it be producing a different number of adults in the end than if you were doing what we did, which was going in and randomly choosing individual larvae to remove. And what you see is kind of a variety of responses. This is Aedes albopictus, and this slope of survivor or proportion surviving versus percent mortality on day 2. And this is proportion out of the initial cohort of 150. It's essentially flat. This would be a case where what you expect to happen for Aedes albopictus is no real change in the production of adults if you impose even 75%, or I think this was 70% mortality on the population. And you see a little bit different pattern for Aedes aegypti. You see here initially, as you impose higher and higher levels of mortality, the numbers or proportion of survivors is going up, but eventually it does seem to come down. And for Aedes triceriatus, it starts out at a very low fraction surviving, but as you impose higher and higher mortality, it just keeps going up all the way to 70% kill on day 2. Notice though the red line. The red line is with the copepod predator. And we couldn't control what fraction they killed, but you can see that they were typically killing somewhere between about 15 and maybe as high as 60% mortality on the mosquito population. In every case, what you get is significantly greater number of adults or greater proportion of survival to adulthood with the real predator than you do with random mortality. And that was quite consistent for all the Aedes. And we wondered about why that was. And I think what's going on here is the predators, particularly these tiny copepods, are probably killing the individuals in the population that are the least likely to survive to adulthood anyway. They're probably the most debilitated, poorly fed stragglers of the population. Killing the weak and the infirm is basically what they're probably doing. And what that means is the survivors they leave behind are going to be the most viable, robust larvae that are most likely to make it to adulthood. So your question's really interesting one. Natural predators probably are going to be in some ways less desirable than any other sort of intervention that could achieve higher rates of kill later in development and knocking out those individuals that are most likely to be robust survivors to adulthood. In that regard, it might well be that something like Toxorynchides would be a better choice, in the sense that Toxorynchides, when they get to be older larvae, can of course take mosquito larvae and pupae, killing right up till the end, whereas the copepods are very size limited. So I think there is potential there for predators to produce this overcompensatory effect. And some data from the field are at least consistent with that as well. Did that get at the question you were thinking of? Of course, the person's on chat, so they can't answer immediately.
She says yes, thank you.
Yeah. The next question is, integrated mosquito management calls for diverse control approaches that target all life stages. Biorational larvicide and biocontrol are two desirable options. With the high variability in field conditions, how can we, as control districts, test whether methods we use could result in overcompensation or other undesirable ecological outcomes?
Mm-hmm. Yeah, and it's another great question, because what you want to know really is not do you have a lot of dead larvae in the water, or not that you have emptied out a certain number of containers. What you'd like to know at one level, ultimately, would be you've reduced the risk of disease transmission. That's very difficult data to get. It's even difficult to get good data on the frequency of infected mosquitoes. One of the reasons that we did that experiment in Rio de Janeiro, or field study in Rio de Janeiro, when we did it was in the middle of a dengue outbreak. And a lot of the data suggests that when you're not in a dengue outbreak, the frequency of arbovirus in the insects is really very low. So it's hard to detect that you've had an effect on risk of disease. All right, well, ultimately you could just look at the production of adults. And that would be an interesting thing to do in the context of the question you've asked, where you're trying to reduce the population of biting adults. But I would argue that what you might want to do is more labor intensive than just using BG sentinel traps and counting the number of adults in the traps. You might want to get a sense of, are we getting bigger adults? If we think that that there's a general trend for larger adults to be more likely to transmit pathogens. Larger adults are also more fecund too. They'll lay more eggs. So that would be another thing to do, is get at some of those traits of the adults that are being produced. And I think another thing that would be useful data to have would be not even just to sample the adult population, but to get, for instance, the egg input to the containers to see if that is trending up or trending down, to look at the developmental progress that you're seeing. So, gathering the rich data across the whole life cycle of the mosquito would be one way to kind of have a whole bunch of checkpoints to see that what you're doing is really producing the effects you want and minimizing the chance of the undesirable effects. And I realize, even though I don't do mosquito control, I realize that what I just said is very labor intensive and probably kind of expensive to do. If you wanted to go for the least costly thing that you would like to do, the closer you can come to getting every last individual mosquito, the better, because it's kind of a truism. If you can kill nearly all of them, you will likely reduce the population and reduce disease transmission. So you were describing integrated control methods. My limited experience with BT, for instance, is if you do BT and do it right, you will kill a huge fraction of the larvae in pools or in containers. I guess more the problem with integrated approach to BT is how you get it into all those cryptic containers that these Aedes would be in. If that sounds like there's no easy answer to this, I think that's probably accurate. It's kind of why people like the idea of these species-specific, high-tech things like gene drive mosquitoes that will suppress a population. It's not only species-specific and well-targeted, but it holds out this prospect that you can do a surgical strike and remove just the organism that you're after with high effectiveness. The monitoring is critical. And one of the things we're interested in right now is we're working with Florida Mosquito Control District near Fort Myers, Lee County, on their SIT program for Aedes aegypti. And one of the things we've been doing is monitoring containers for Aedes albopictus and looking at the traits of the Aedes aegypti that are being caught in the field. So that's something, that kind of monitoring, I think, is one of the ways that you get an idea that you're not producing those undesirable effects.
Okay, thank you. I'm checking the chat. I don't see any more questions.
Okay.
Just scroll. Nope, I think you answered them all. So once again, I wanted to thank everyone for coming. I placed 2 links in our chat today there was one link from the San Gabriel Valley Mosquito and Vector Control District. They sent a link. It demonstrates how a germ, in this case, the West Nile virus, travels through your body, through the body of a mosquito and into an unsuspecting host. They use that for grades 4 through 12 when they're doing outreach in schools. So that might be something everyone could use if they're doing the same kind of outreach. I also put a link to our rodent control survey, which will help us gain knowledge from professionals and help us tailor our educational offerings regarding rodent control. So if you're new to our webinars, please take a moment to do the survey. Just once again, thank you everyone for attending today's webinar. Please email us at education@nola.gov if you want to be placed on our mailing list. If you have any other questions that you think of, you can email them to education@nola.gov and we'll be sure to get the answer to you. And if you have any suggestions for future topics, you can email us also at education@nola.gov. Our next webinar will be on March 10th, and it is Urban Rodent Control Seminar, and the topic will be the importance of rodent management. So once again, that's March 10 at 2 p.m. You can email us at education@nola.gov if you want to be placed on that list. But once again, thank you for the great lecture on Aedes and the competition. Really appreciate it.
Thank you. It was a pleasure, and I really appreciate the invitation.
Thanks, Steve.
Okay.